Have you ever wondered how scientists solve mysteries inside of our cells? Well, I have too. Today, we're going to be diving into a fascinating tool called the Amplex Red Assay, a molecular detective of sorts that helps researchers track down a sneaky suspect, hydrogen peroxide. What is hydrogen peroxide? Hydrogen peroxide is a double edged sword. In your bathroom, it's that brown bottle for cleaning cuts. But inside of our bodies, it's a tiny cleaner, breaking down harmful stuff and sending important messages to our immune system. However, it can be helpful and harmful. Too much of it can damage cells, like rust on a pipe. But in small doses, it's a signal that keeps things running smoothly. Scientists need to be able to measure it to understand diseases like Alzheimer's, Parkinson's, or diabetes. But how do you track something nearly invisible? This is where our detective comes in. Meet our Amplex Red, a clever chemical detective of sorts. You can't see hydrogen peroxide, but you have a special tool. Think of a light bulb that only turns on when a specific type of gas is present. That's kind of what Amplex Red does. It's a special chemical that stays invisible and colorless until it comes into contact with hydrogen peroxide. But there's a catch. Amplex Red can't do this job alone. When hydrogen peroxide is around, Amplex Red teams up with an enzyme partner called HRP. Think of it as its sidekick enzyme, if you will. HRP acts like a matchmaker or a key that unlocks the reaction. When hydrogen peroxide is around, HRP grabs it and uses it to activate Amplex Red. The reaction is exact. For every single molecule of hydrogen peroxide, exactly one molecule of Amplex Red gets changed. Together, they transform hydrogen peroxide into something we can see, a glowing red molecule called resorphin, as exemplified on the bottom of our screen here. The more hydrogen peroxide there is, the brighter the glow. So the further left we go on the bottom of our screen, the less hydrogen peroxide there is. Therefore, the brighter the red, the more hydrogen peroxide there is. So how exactly does this assay work? How would we use it in real life? Here we go. So let's break it down with an example. Imagine scientists studying worms to learn about aging. Here's how they would use Amplex Red. Step 1, they expose the worms to something stressful, like a harmful bacteria. Step 2, they wash the worms and then add them to a tiny well with Amplex Red and HRP that you would find in your laboratory, just like this. Then you add your detective and your sidekick. And if the worms release hydrogen peroxide, then the mixture glows red. A machine will ultimately measure the glow like a light meter for clues. The brighter the glow, the more hydrogen peroxide the worms made. It's like a burglar alarm going off when the culprit is ready. So why does this matter? By tracking hydrogen peroxide, scientists can test new drugs to see if they reduce harmful stress in cells, study diseases where hydrogen peroxide runs wild, like Parkinson's, even learn how our bodies fight infections. It's like basically giving researchers night vision goggles to see what's happening inside living things. So next time you see glowing red liquid in the lab, remember, it's not magic, it's science. The Amplex Red assay is just one of the many tools helping us crack life's biggest mysteries, one molecule at a time.